My name is Nick Brookhouser, and I'm a PhD student at Arizona State University. My research entails engineering human-induced pluripotent stem cells to investigate the onset and progression of Alzheimer's disease. Alzheimer's disease is a devastating neurodegenerative disease that has a tremendous socioeconomic burden on patients and caregivers alike. Alzheimer's disease affects over 5 million individuals in the United States and has a direct costs estimated in excess of $200 billion per year. Prevalence in the United States is expected to rise to an estimated 13.8 million individuals by the year 2050, representing a major public health concern. Although a majority of cases are sporadic, several genetic risk factors have been identified. Of these risk factors, polymorphism in the apolipoprotein E or ApoE gene, a lipoprotein transporter involved in cholesterol metabolism, is the strongest and most prevalent. Although it has been established that ApoE modulates the formation of amyloid plaques and neurofibrillary tangles, two pathological hallmarks of Alzheimer's disease, the underlying mechanisms of how this risk is conveyed has yet to be uncovered. A lack of mechanistic understanding, as well as a failure of potential therapeutics in clinical trials, can in part be attributed to the over-reliance of animal models of Alzheimer's disease that, in addition to ethical considerations, do not display important pathological hallmarks of the human disease and have not adequately modeled the complex genetics associated with sporadic Alzheimer's disease. My project utilizes two transformative technologies, human-induced pluripotent stem cells and CRISPR-Cas9 gene editing techniques, to investigate the mechanisms by which ApoE variants contribute to disease progression. With human-induced pluripotent stem cell technology, it is possible to obtain a fully differentiated cell type, such as a skin cell, from an Alzheimer's disease patient and reprogram it back into a cell type that is capable of differentiating into all cell types of the mature adult body, such as neural cells of the cortex. Over the past year, I have developed a diverse set of induced pluripotent stem cell lines from Alzheimer's disease and non-demented control patients with varying ApoE genotypes, and developed a three-dimensional organoid-based culture model that better mimics the architecture and complexity of the in vivo cortical tissue, as compared with two-dimensional culture models that are currently being used. With current gene editing techniques, I have been working to establish isogenic ApoE-induced pluripotent stem cell lines that differ only in the ApoE gene. This allows us to study the effects of a single genetic change while controlling for the genetic background of the patient. By using these isogenic induced pluripotent stem cell lines in a three-dimensional cortical neuronal culture model and comparing expression profiles using a technique called RNA-seq, we are identifying unique gene expression profiles that are independently defined by ApoE genotype and disease status. Future bioinformatic analysis will reveal candidate genetic, biochemical, and signaling pathways that will provide a more definitive relationship between ApoE genotype and Alzheimer's disease onset and progression. Overall, the use of pluripotent stem cells allows us to address many experimental questions that would otherwise require numerous animals to examine, and offers a simplified and accessible system for the study of genetic risk factors as well as drug screening. Finally, I want to thank the International Foundation for Ethical Research for the support of this work, and thank you for listening.